Okay, Assalamualaikum and good afternoon everyone. So we will continue a bit on protein metabolism. Okay, so in this chapter, it's basically for us to understand what happens to protein once we have digested them in our body, and then what will happen. Let's say after the metabolism, we have waste product. How do we cater those waste products? Will it be stored in our body or will it be eliminated? If it's eliminated, then how would we eliminate those waste products, for example? And then a bit on ketogenic and glucogenic amino acid, just for you know that all of these metabolism are connecting to each other, meaning that carbohydrate metabolism, we have looked through, it relates to protein metabolism as well. Okay. Okay. Very briefly, a recap of the importance of protein. So all the proteins that we have digested from our diet or being synthesized in our body are very important. First thing is for our cell integrity. Okay. And then another important thing would be the source of nitrogen. So if you remember the structure of amino acid, we have amino group, carboxyl group, right? So this amino group contributes to the nitrogen, okay, that are being needed for other type of metabolism. Okay, and then dietary protein also serve as, for example, the amino acids, okay, for our body proteins, and then the carbon molecule in the amino acid can be oxidized to yield energy. So you will see the relation of amino acid and also Krebs cycle. So Krebs cycle is the one we see in carbohydrate metabolism after glycolysis. So you will see also how this amino acid can contribute to that. Okay, and then carbon and nitrogen atom can be used to synthesize other nitrogen containing cellular constituents. For example, our receptor, our enzyme. So they will require these carbon and nitrogen atoms to build up them. Okay. Okay. So before we go into metabolism, this is just a recap for us to see the digestion of protein. So you need to remember digestion of protein starts in our stomach. If carbohydrate, it starts in our mouth, right? Our saliva, for example. Okay, but for protein, it will start in the stomach. So our food will enter our stomach and then it will stimulate this hormone called gastrin. Okay, so for hormones in carbohydrate, it's insulin and glucagon. But for protein, the ones that related to protein metabolism would be gastrin. So what will this gastrin do? It will stimulate release of gastric juice. Okay, so gastric juice, because it's a juice, it's a combination of things. This gastric juice contains hydrochloric acid, pepsinogen, and renin. Oops. Okay, so pepsinogen and renin is one of the enzyme that can withstand a very low pH. Okay, it can survive a pH as low as 1.5 to 2.5. So it's kind of different from other enzymes, which may require a more neutral pH. Okay, but for the ones inside our stomach, it can withstand a very low pH. Okay, so what will happen? This gastric juice is very important in terms of its helping to denature the protein. Okay, those acidic environment will denature the protein, and this HCl can also provide serve as an acidic environment for pepsin to be activated. Okay, so this pepsin, a type of enzyme, is actually secreted as pepsinogen. So the hydrogen ion from the hydrochloric acid will activate this pepsin. So what will this pepsin do? Because it's an enzyme, it will cleave the peptide bonds of aromatic amino acids and acidic amino acids. Okay, so basically the general function of pepsin is actually to cleave the peptide bonds of amino acid to make it into smaller chunks. So if you could imagine, so let's say this is our protein from our diet, a piece of our chicken, for example. So this chicken will be broken down into peptide chains. So there will be a lot of peptide chains. Okay. Maybe due to the fact that hydrochloric acid is breaking them down, and then what will happen? This pepsin will come and break this peptide bond. So now we have a single molecule of amino acid. Okay. Okay. Other than pepsin, we also have rennin. So this rennin can help infants to clot milk, especially, okay, and convert milk to a more solid form, okay, to prevent rapid passage of milk from the stomach. Okay, so this rennin, when we drink our milk, so it will clot them, okay, to make it slower to be broken down. Okay, so that it wouldn't be just passing through our stomach. Okay. I lost my mouse. Okay. So this is just a recap for you. This is not the important part of protein metabolism. Okay, this one you will learn in if you take physiology later on. Okay, so but this is just for you to understand. This has to happen first, meaning that this protein has to be broken down into amino acid first. Only then it can undergo metabolism. Okay, so digestion first, followed by metabolism. Okay. Okay, before we go through the cycle of protein metabolism, okay, this is something that everyone should take note of. Our amino acid inside our body, we don't have storage facility. Okay, for carbohydrate, the storage form of carbohydrate is glycogen. Sorry. So the storage form of carbohydrate is glycogen. Let's say we have eaten our bread or our rice. So after some time, after we have eaten, only maybe 50% will undergo metabolism for energy. The other 50% is not required. So what will happen? Our body will save this carbohydrate as glycogen. Next time when we are fasting, for example, our body will break down this glycogen. Okay, but it's different with protein or amino acid. So amino acid is not stored in our body. Okay. So in general, this amino acid will be incorporated into functional protein, meaning that they will be used as building blocks to make protein. Okay. And this cycle is called as amino acid pool. Okay. I will show you the equation. Okay. So basically, this is what happens. When we say nitrogen balance, it is equal to amino acid balance. So meaning that the amount of amino acid that is being ingested, taken in our diet, for example, okay, will be equal to the amount that is being secreted in our urea. Okay, so we don't store our amino acid. Let's say, this is just to give you a number to illustrate. Let's say we have eaten 10 amino acid, then 10 would be used by our body. Let's say we have, macam ni. Let's say our body require, let me draw lah. Okay, let's say our body require 12 amino acid in our daily function. But you have eaten, for example, 15. Let's say that day you, sorry. Okay, you have eaten 15. But your body only require 12. So what will happen to the balance 3? It will be secreted out. Okay, so that is what meant by we don't store our amino acid. Okay, so so this is what we call as amino acid pool. The amount of amino acid going in and going out is at equal rate. Okay. So in healthy adults, if you are healthy, you don't have any medical condition, the total amount of protein will remain constant. Okay. And the rate of protein synthesis is just sufficient to replace the protein that is degraded. Okay. So this condition, we call them as protein turnover. Okay. So in healthy adults, protein turnover is constant. Turnover means the amount in and the amount out is same. Okay, but in infants, okay, for kids, for example, or babies, the turnover is high. Okay, because they are still building their tissues. especially in kids, they are still growing, so their body is constantly require protein or using protein. Okay, so their turnover is high, but it will eventually decreases with age, okay, due to the age factor. Okay, so this is the overview of protein metabolism. Okay, so the ones that is in the box would be the enzyme that is involved. Okay, the green colored one, we have aminotransferase, for example, okay, glutamate dehydrogenase. Okay, and the background over here, if you see, this is skeletal muscle. This is to indicate skeletal muscle. Okay, on your left would be liver. So protein metabolism, if you want to do it in the box form, like I have asked you to do in carbohydrate, okay, for carbohydrate, for example, glycolysis, you have localization in cytoplasm ke, in nucleus ke, okay, in liver. So for protein, we can do the same thing as well. So location, you can say in skeletal muscle and also in liver tissue. Okay, and then we will look into detail in liver, which part of liver, in the cytoplasm or in the mitochondria. Okay, so this is one thing that you can start to fill in the box. Okay, you can create the box and then just write protein metabolism. You can state okay, location where and so on. All right. Okay, so let's start for the first one. Okay, so the first step in protein metabolism, it happens in the skeletal muscle. Okay, skeletal muscle tissue. Okay, this is just an example, alanine and alpha ketoglutarate. So alanine and alpha ketoglutarate is a type of amino acid, okay, that is always present inside our skeletal muscle. So I'm just picking this as an example for you to see what happens in the cycle. Okay, later on we will try to change this amino acid for you to see the process. Okay, so again, protein metabolism starts in the skeletal muscle. It can start with any type of amino acid. For this case, I choose to show you the example of alanine and alpha ketoglutarate. Okay. Okay, so what will happen? The first thing, this alanine and alpha ketoglutarate will react together, and this reaction of alanine and alpha ketoglutarate is catalyzed by aminotransferase. If you see the arrow that I have drawn here have two sides, meaning that this reaction is reversible. Okay. Okay, so alanine, okay, will react with alpha ketoglutarate and they will produce two new molecules named pyruvate and glutamate. Okay, so this whole process is called transamination. Okay, I want you to focus on the changes that happens over here. Okay, we have alanine and also alpha ketoglutarate. So this aminotransferase enzyme, they will catalyze the switch or the transfer between these two groups. You can see my mouse, right? T. You can see my mouse moving. Okay. So this aminotransferase, they will catalyze the transfer between amino group of one amino acid and the carboxyl, the oxygen from the carboxyl group of another amino acid. Okay, I'll repeat. Aminotransferase is catalyzing the process of transamination. So what happens in transamination? Amino group from one amino acid will be transferred to the oxygen group from the carboxyl of another amino acid. So if you see, there's a switch in place over here. So now this amino group of alanine will get this oxygen group from alpha ketoglutarate. Okay, and this alpha ketoglutarate will get this amino group from alanine. Okay, so the new molecule that is formed now after the switch happened is pyruvate. Okay, if you see this pyruvate, no longer amino group, but we have the oxygen and glutamate. Okay, so this is the new molecule. So let me repeat, for protein metabolism, first happens in the skeletal muscle tissue. So the first process is transamination. So what happens in transamination? There's a reaction between two amino acid catalyzed by aminotransferase. So this aminotransferase, what will it catalyze? It will catalyze the transfer between amino group and oxygen from the carboxyl group from two amino acid. And it will form new molecules. Okay, so this is the first step, transamination. Okay, so this is just to show you, okay, if you remember, pyruvate is the end product of glycolysis. We will start with, for example, glucose 6-phosphate or glucose, and then all the 10 steps of reactions that happens. Lastly, in glycolysis, we will get pyruvate. Okay, 1 molecule of glucose, 2 molecules of pyruvate. Okay, so this pyruvate will be used for example, it can enter the Krebs cycle to produce ATP, or in anaerobic condition, it will be formed lactic acid. Okay, so this is to show you in protein metabolism, we can still generate energy. How? Because of the formation of pyruvate. Okay, this pyruvate coming from where? Coming from amino acid. Okay, alanine. So this pyruvate can enter the Krebs cycle to generate an energy in the form of ATP. So that will happen. What will happen to pyruvate? So what will happen to this glutamate? This is just the written form of this. You can read this later. Okay. Oh, okay. Before we go to what happens to glutamate, okay, the standard flow, or what we call as the fate of this pyruvate, if in aerobic condition, means got oxygen, it will enter the Krebs cycle just now. Okay, but in anaerobic condition, it will form lactic acid. So what will happen to this lactic acid? This is just an added information for you. Okay, so this lactic acid will go to the liver, transported to the liver, and then it can undergo gluconeogenesis, patah balik. Okay, reverse back into glucose. Okay, and then this glucose can undergo glycolysis again and then produce ATP. So that is how protein metabolism in one way can help to produce energy. When in aerobic condition, it will enter Krebs cycle, but in anaerobic condition, it can produce lactic acid, which will be used in gluconeogenesis to produce more glucose. Okay. All right. So the first step just now is transamination. Okay. So in this case, two molecules of amino acid is two molecules of amino acid react and produce pyruvate and glutamate. So what will happen to this glutamate? Glutamate will be transported into the liver. Okay, from our skeletal muscle, let's say from our hands. So now our body will transport them to the liver. So what will happen inside the liver? Okay, so this is the second process. So just now is transamination. Now this glutamate, after being transported to the liver, it will undergo oxidative deamination. Okay, so this oxidative deamination, what will happen? So glutamate will be converted back into alpha ketoglutarate. So if you see just now, this glutamate is coming from alpha ketoglutarate as well. So in oxidative deamination, it will remove this amino group, and because it's oxidative, meaning it involves oxygen, oxygen group will be added back. Okay, so in oxidative deamination, basically the word deamination means to remove. Remove what? Amine group. Amine group is the amino group over here. So deamination, now it's kicking off this amino group, kick off and replace with oxygen. Okay. So where will this amino group go after being kicked off? It will form ammonium ion. Okay, so this ammonium ion is very toxic. So this is where our body will try to eliminate ammonium ion. Okay, so again, as I showed you earlier on, transamination over here, trans mean to transfer. Transfer who? Amino group. Okay, amino group from one amino acid to another. Okay, while oxidative deamination, deamination means to remove amino group. Kick off the amino group, and it involves, because it's oxidative, it involves oxygen, so it will add the oxygen group back to the structure. So if you see glutamate and alpha ketoglutarate, the difference is in this green circle. Okay, so now the amino group that is being kicked off or removed, okay, don't write kick off, okay, in your exam, write remove. Okay, this amino group will form ammonium ion, which is toxic to our body. Okay, the enzyme involved in oxidative deamination would be glutamate dehydrogenase. Right? Can? Can. You can draw. Drawing also can, as long as you write it properly lah. Don't just draw and leave it like that, meaning you have to mention this is what process, this is what enzyme. If your process is self-explanatory, meaning that if I see, I can understand. You may need to put numbers ke, this starts first, this and then second, and then third, macam tu. Better like that. So if you want to draw, make sure your drawing is and then the flow. I would suggest if you want to draw and write the description. Draw and write. Tell me the flow. Okay. Okay. Again, so this is just the written version of oxidative deamination. Okay. So now let's focus on this. So after oxidative deamination, what will happen? Okay, so this ammonium ion, because it's very toxic, okay, it will be secreted as urea in our urea. Okay. Okay, so before we go into urea cycle, okay, just to share with you, for example, if we have another type, so in this case, sekejap macam ni. Okay, so now after oxidative deamination, again we will get the alpha ketoglutarate. So what will happen to this alpha ketoglutarate? It will then react with another type of amino acid, just like when it reacts with alanine. So meaning that after the formation of alpha ketoglutarate in the liver, our liver will transport them back to the skeletal muscle, for example, to react with another amino acid. Okay, for example, like this one. So this alpha ketoglutarate can react with, for example, aspartate. The same enzyme, it will aminotransferase. So what will happen? It's the switch between amino group and also the oxygen. It's the same thing as alanine just now, but I am changing alanine example with aspartate. So aspartate is another type of amino acid. So it can react, alpha ketoglutarate and aspartate can react together, catalyzed by the same enzyme as transamination earlier on, aminotransferase, and then form another two molecules. For example, in this case, glutamate and oxaloacetate. Just now, if alanine react with alpha ketoglutarate, we have pyruvate and glutamate, right? So if aspartate react with alpha ketoglutarate, we will form oxaloacetate and glutamate. Okay. So this oxaloacetate, if you recall from your Krebs cycle, it is one of the intermediate in Krebs cycle. So let's say this is the Krebs cycle. We have a lot of intermediates in between. Intermediates is the molecule. So oxaloacetate is actually one of the intermediate in the Krebs cycle. So this oxaloacetate, again, it can enter because it's in the Krebs cycle, it can support the Krebs cycle and form more ATP. So if you remember, Krebs cycle is about generating ATP. So this is how protein metabolism contribute to your energy. Protein can still give energy. How? Because it's producing intermediate of Krebs cycle. And then this glutamate will undergo oxidative deamination again to remove this amino group as ammonium ion. Okay, so this is just to show you the example. Okay, so this is the oxaloacetate I mentioned earlier on. It's part of this Krebs cycle, the citric acid cycle. So if more protein that is being ingested, and then they can produce oxaloacetate, then it can enhance our citric acid cycle and enhance the production of ATP. Okay. Okay. Even alpha ketoglutarate is actually one of the component in the TCA cycle. All right. So let's go back to this ammonium ion. What will happen? So our body will dispose ammonium ion or this ammonia in 3 ways. Okay. The first one would be this ammonium ion will combine with alpha ketoglutarate to form glutamate. Okay. Or this ammonium can combine with glutamate to form glutamine. If you remember, glutamine is a type of amino acid too. Or this ammonium ion can go through urea cycle to form urea. So there are 3 ways our body remove this ammonium ion. So the idea is that this ammonium ion cannot exist as ion. So ammonium ion cannot exist in this state. It needs to be combined with another molecule so that it will be safe. If it's in this ammonium ion state, it is toxic to us. So how our body do it? It will let this ammonium ion combine with another amino acid, for example, and form another amino acid. Okay? Or it can be secreted or excreted through the urea cycle. Okay. So this is what happens if ammonia is built up in our body. We call them ammonia intoxication. Okay, meaning toxic. So it can impair the function of our brain. It can cause tremor. So this is just to share additional info for you. This ammonia, they can go into our brain cells, and then it can, when ammonia ion enter our brain cell, it will bring along water. So it attracts water to go in. So what will happen when it's being accumulated in our brain cells? Water will also go into our brain cells. Our brain will start swelling, jadi bengkak, because of the amount of water that is too much. So when our brains become swollen like that, it can cause nerve damage. Okay, that's why we have all of these symptoms. Tremor tangan jadi ketar-ketar because nerve is damaged. Okay, convulsions, nerve is damaged. Even nausea and vomiting because it's affecting our nerve. Okay, it's causing all of these symptoms. Even blurred vision, especially if the nerve is directing to our eye. So this is the danger of ammonia intoxication. So our body needs to excrete them. Okay, so this is just the written version. You can read this later. I'll show you the diagram. Okay, so this is the overview of urea cycle. So again, urea cycle is very important for our body to eliminate the ammonium ion. Okay, and it can also form arginine and fumarate. Okay, which is important for nitrogen metabolism. Urea cycle. Sorry. Okay. So urea is different from urine. So urine is from your kidney, kan? Urine lah, okay? But urea is from your protein, okay? Kidney, protein metabolism from liver. So it's different thing, okay? So what will happen? This ammonium ion from here, okay? This ammonium ion, it will react with two molecules of ATP and also carbon dioxide that is already present. Okay, and then this reaction happens inside the mitochondria, inside our liver cells. So this we are going zoom into. So from the liver itself, we zoom into each of the cell. So each cell has its own mitochondria. So inside the mitochondria, ammonium ion will react with two molecules of ATP. Okay, if you remember back, chapter 2 is about cellular biomolecule. You learn mitochondria produce ATP, right? So this ATP is coming from the mitochondria and also carbon dioxide molecule. So these three molecules will react together and form carbamyl phosphate. So this is a new molecule that is formed. So the enzyme that's catalyzing this reaction is carbamyl phosphate synthetase type 1. This is how you remember it because it's synthesizing, it's producing carbamyl phosphate. The enzyme is synthetase, carbamyl phosphate synthetase. Okay, type 1. Okay, and then this carbamyl phosphate, it will go out of the mitochondria inside the cell bag. Okay, carbamyl phosphate go out of mitochondria, still inside the liver cell and enter urea cycle. Okay, this is the actual urea cycle. Okay, the same as TCA cycle. It has a number of intermediates. So the purple ones are the intermediates. The ones in the blue box are the enzyme catalyzing the conversion into new intermediates. Okay, so from ornithine, for example, it can be converted into citrulline. Then citrulline converted into arginine succinate. Okay, and so on until eventually it will form arginine. So this arginine, okay, it can remove, this arginine will be converted into ornithine, but at the same time, it will remove a urea group. So this urea, okay, will be secreted in our urine together from the waste cycle, waste product from our kidney. Okay. Okay, let's stop for a while, about 2 minutes. Try to digest first because the Zoom time is almost ending. Then we come back to the same link, okay?
Okay, doctor.